My name is Cailin Collins, and I'm a postdoctoral fellow in the Majeti Lab at Stanford University. Today, I'd like to tell you about my work harnessing the power of genetic engineering to putting a stop to the development of acute myeloid leukemia. If you take a moment to think about the human body, each one of us is absolutely remarkable. We are made of 37 trillion cells working together to perform thousands of different tasks that allow us to go on a walk with our dog, enjoy a delicious meal with our family, and even remember memories from our past. And each one of these cells started from a single cell at the beginning of our lives, because that cell held all of the information it needed to form our impossibly complex bodies within its DNA. The magnitude of this reality is as unfathomable as the expanse of the universe, and has fascinated me since I was a little girl. I can remember the first independent scientific question I asked. It was when I was about 7 years old, and I had a case of the hiccups that would just not go away. And I wanted to understand what it was that was causing these uncomfortable convulsions. So of course, I went to our bookshelf, because the internet didn't exist at the time. And I pulled the volume H of Encyclopedia Britannica off the shelf and learned that hiccups were the result of an irritation of an organ called the diaphragm. Now, there wasn't any answer as to how to get rid of the hiccups, so I had to resort to a scare from my sister. But I thought it was so cool that someone had figured out how the diaphragm normally works, and then in that way was able to explain how and why things went wrong with a hiccup. Fast forward 30 years, and I'm completing my training as a physician scientist, and I remain fascinated in understanding how and why things go wrong in the body, because I want to be able to stop these things from happening before they go wrong. My interests have honed in on what I think is the quintessential process of cells going awry, and that is cancer. I'm specifically interested in a type of blood cancer called acute myeloid leukemia, or AML, which is one of the most aggressive and deadly forms of cancer, with an only 30% overall survival rate, which drops to lower than 5% in patients above the age of 65. This interest brought me to Stanford to train clinically as a hematologist and oncologist and continue my postdoctoral research with Dr. Ravi Majeti, studying the earliest stages of development of AML. Through decades of genome-wide studies, we now know that AML develops as the result of an accumulation of mutations over the course of our lifetimes. Unlike other cancers, each patient's AML is the result of a relatively few number of mutations, on average only 5, which compared to, say, hundreds in things like colon cancer or melanoma is quite small. This fact presents us with an opportunity to study the biology underlying the development of AML, because we know that only a few mutations are necessary to result in a full-blown cancer. For years, we've studied the contributions of mutations to disease by studying these changes in genetically engineered mice or by transiently changing genes in human cells. However, we are at a very exciting time in biomedical research because we now have the ability to directly edit genes within a human cell using a technology called CRISPR-Cas9 gene editing. This technology was actually discovered in studying how bacteria defend themselves from infections. And in 2020, 2 female physician scientists were awarded the Nobel Prize in Chemistry for their contributions towards its development. You are likely familiar with CRISPR because it's made headlines as the technology being used to cure genetic diseases such as sickle cell and hereditary blindness, and also for the controversial spotlight it's gained in the possibility of genetically engineering babies. But for the purpose of my work, CRISPR is just a truly remarkable technology that is opening doors to allow us to ask questions and perform experiments that were never possible before. CRISPR works by using a small RNA molecule to guide the Cas9 protein to a specific spot in the genome, where it then acts as a pair of molecular scissors to cause a double-stranded break in the DNA. We then provide the cell with additional DNA that it can use as a template to repair that damage. And in this way, we are able to introduce mutations that we see in patients into the cells in the culture dish. I am using CRISPR editing of human blood stem cells to study the earliest stages of development of acute myeloid leukemia. By combining 2, 3, or even 4 mutations into a single cell, I can study the essential pathways that occur as these cells sequentially acquire these mutations. I then have the ability to study these cells by transplanting them into mice and observing their behavior over time. These models give us the opportunity to test new therapies targeted at the pathways that we discover using our edited stem cells. With these powerful tools and with the continued study of early AML detection in the clinic, I am confident that within my lifetime, we will have a treatment that is able to stop AML before it ever develops and save thousands of lives each year.